I'd now like to welcome Pardis Sabeti, professor at the Center for Systems Biology at Harvard University and a member of the Broad Institute of Harvard and MIT. Dr. Sabeti, thank you for joining us for this Time 100 talks.
Thank you, Alice. I'm really pleased to be here.
Infectious disease experts like yourself have been warning about the threat of new viruses, new bacteria for decades, maybe even centuries. And yet every time one of them sweeps through the world, we seem to be caught unprepared and even a little unaware. Are we more prepared today than we were during the last global outbreak of an infectious disease?
Certainly. I mean, I think that we've come a very, very long way, even since the 2014 Ebola outbreak, and then Zika after that. With each one, we've been learning. We haven't been going as fast as we should, but we've definitely been getting better. And the technologies have been advancing at every step. We're at the point where we're generating thousands and thousands of genome sequences of the virus very, very quickly. New diagnostics are emerging every day. There's a lot of really things to be pleased about. And there's also a lot of things to make us realize that we have not invested enough.
When you look at where we are today in fighting infectious diseases, what technologies or tools do you think we should be prioritizing? What could make detecting and controlling epidemics more effective and more efficient?
There's a number of new technologies coming out from, one that I work on is CRISPR, sort of the gene editing tool. There's also some really excited tools from synthetic biology and nanotechnology that are really transforming the way we can do diagnostics.
Sorry to interrupt you, but could you just explain how CRISPR works?
Most people have heard about it because of this idea of the transformations it can make to human health. It's a gene editing tool. So people think about it as ways of correcting human diseases or the ability to essentially change our DNA. And it can be used in medicine and research very, very well. But fundamentally, CRISPR was originally identified as a bacteria's immune system to viruses. It's how bacteria detect and destroy viruses. And so it's a really, if you think about how things are used in nature, it's designed as a diagnostic, as a way of identifying if a virus is present. And it can also is also being used, and we and others are working on it towards therapies as well, because that is its function, and it does it quite well.
Can you talk a little bit about how a CRISPR-based therapy might be useful against a virus like SARS-CoV-2, the one behind COVID-19?
So there are a lot of different therapies out there, and the kinds of therapies people are interested in are ones that are already available, and that's why there's a lot of excitement about chloroquine and remdesivir, different kinds of antimicrobial therapies that could be used. And then vaccines are another way of preventing new infections from happening. And there's these things in between. So it's sort of like what we do when an outbreak starts is we grab everything off the shelf that we have and try to use all the things like remdesivir we have and see does anything work. We're working to develop a vaccine that's going to take time. It might be a year and a half away. And the kind of question is, do we have therapies now that we can bring new therapies? So like in that middle spot. Jim Crow explains this. That's part of where he works and try to develop different antibody therapies. Antiviral therapies based on CRISPR are really new and have just really kind of been rolling out very recently. So in a study that we published in Catherine Freehe and Cameron Mirabold in my group published a paper to show that this CRISPR even in mammalian cells can knock down viruses. And they ran it against a lot of different viruses, including flu. And for example, in flu, they showed that you could reduce infectivity 300-fold. And so an idea is, it could really help in response to infection, and it may not itself clear the infection, but it may give you enough time. So reduce morbidity and mortality by giving you enough time to respond. And there's still a lot of things to get from here to there to get it. So we safely can see it working. It was in nature, it was supposed to be in bacterial cells. We can see it working in mammalian cells and it seems to not have any off-target effects or things we're concerned about yet. But the next question is, getting it in vivo, getting it in animal models and humans, and showing it can safely be used. And there's a lot of other things from here to there. But as everything is happening in COVID, everyone is moving very, very quickly to try to roll all of these technologies out.
And doing that in real time, developing, taking advantage of new technologies and trying to develop new ways of fighting a disease as it's unfolding. It's not something that you're totally unfamiliar with. You played a really important role during the Ebola epidemic several years ago in quickly sequencing the virus behind Ebola from patients who'd been infected. And you were doing it in real time as the outbreak was unfolding. And that idea of sort of learning as you go and applying that knowledge back to potentially helping to control the epidemic is relatively new. Can you tell us some of the lessons that you learned in doing that? And do you see some of those lessons learned from that experience now being applied to COVID-19?
The fact that we could do it so quickly had a lot to do with the technology was just kind of coming up. It was coming to the point where it wasn't going to take 5 years to sequence a genome. You know, you could sequence them quickly, and we had been working on other viruses, related viruses for some time, and my group had actually been studying Ebola as well. So we were positioned and we had a lot of technologies in place. We'd published a paper 2 years before the Ebola outbreak hit, where we actually drew a picture of the Ebola virus into the Congo River and said, you know, these viruses are here. You know, a lot of your best insights are just in the field. By being in the field in West Africa, we were just realizing that there are so many of these infections that were just going undetected because we didn't have means to detect them. Even in the United States, you know, here in New England, there's a virus called Powassan virus that infects a lot of people, and there's just no way that people have to detect them. So you don't know if you have it or not. And one day when it becomes an outbreak, we'll be surprised, but we don't have to be if we had better means of detecting them. So anyway, so in that case, we were already poised and ready to do that. The release of the data was an interesting one for us. It was this interesting thing because I got the amazing time recognition for essentially deciding I didn't care about recognition. And so it was this interesting moment for my group because we got this tremendous recognition in the back of tragedy. So in one way it was very conflicting because it was like, you don't want to, you know, have like success at a time when so much is wrong. But the only thing that we're proud about is that we got recognition for saying, as soon as we generated the data, we just released it to the web. And assume that that's what you should do in an outbreak, but there's not time to lose. And so that idea that, you know, publish the data first, let everybody look at it, and then think about publication later was sort of, for us, something that was natural to do. It broke a lot of barriers, and we made a lot of collaborations around the world in the process. And I'm really grateful to see that so much of what's happening now, you know, there's so much cooperation happening. There's also a lot of not cooperation happening. You know, there's the good and the bad, but there's a lot of good people out there showing solidarity and working together.
And in some ways, as you said, your decision to publish that data and have it out there, we saw that happen very quickly here during the COVID pandemic, right? And where we saw some of the scientists who had seen the very original cases in China very quickly within weeks published the genomic sequences from the samples of those very first patients. And so the world was then able to say, you know, groups here start working on a vaccine, a group over here working on the antiviral treatment. And that was really a game changer. Can you talk a little bit about how valuable having the genetic information or the genetic fingerprint of, you know, SARS-CoV-2 in this case, how valuable that is?
It is absolutely necessary. Not sufficient, but absolutely necessary to make things move, right? So the genome sequence of the virus is its blueprint. It's what we use to target diagnostics. It's what we use to target therapies and vaccines. So it's critical information that you need to share right away. And what was really tremendous, and what I wrote about in Time, thanks to you, you know, right as the outbreak started, was just how unique this event was, that within likely weeks of the virus emerging in the human population, they'd already identified it and had the genome sequenced very shortly after. So we were in a really good position as a global community to know about this virus. The genomes were generated and published and made available. I mean, there obviously are some things that slow things down in the process, and I'm sure that there's a lot of consternation on that side, but fundamentally, this was very, very fast, one of the fastest we've seen. At the same time, this virus was moving very fast. It's a coronavirus, they're respiratory viruses, and they're known to be highly infectious. And so there wasn't a moment to lose. And where, you know, what's unfortunate is we lost a lot of ground that we had. We had all this information, but diagnostics just took so much longer to become available to the community. our group and other groups around the world had immediately, as soon as the genomes were published, had working diagnostics in our lab. And every hospital in the country could have a working diagnostic in the lab, because PCR and other things are things that anyone could just prop up where they are. But it's unfortunate because we haven't thought about that enough. Like the next time a new virus comes, we can't have it be another few months before hospitals can get tests. And that is one of the kinds of things that needs to be transformed, the sort of the culture and the infrastructure around how we do this. Because the data is available, the technology is available. It just has to be the process that changes.
So that's one of the lessons that we hopefully should take from this experience, is that when the next one does hit, and as experts like yourself keep telling us that we will have another outbreak from some novel virus or bacteria, that we need to have a better system for being able to test people quickly. So you mentioned that it's not the technology, so the science is there. It's sort of like, a matter of making sure that the rest of the process, making sure that it's safe, it's effective, and scaling it up is ready and up and running. What are some of the biggest holes there that you see that we could be improving on to make sure that the next time the testing doesn't fall so far behind?
I think one of the things is essentially that having it so that every hospital in the country would be poised and positioned to immediately stand up a lab developed test in their clinical labs, right? So that a new virus is put forward, everyone has the practice and the comfort level to just say, I'm gonna put in a test in place. Like while other commercial tests are being developed and maybe, ones that are higher throughput or easier to use, that all should be happening in parallel, but like a lot of these things should be happening in parallel. What was really frustrating for us is to watch so many people just waiting for a test to become available. And fundamentally in an outbreak, every single actor, every person should be able to do something. Like they shouldn't have to just wait for others to help them. It's one of the reasons why we and others are developing mobile applications that connect people and that allow them to share information in real time as well. We and healthcare workers. So there's a lot of things both on a diagnostic level and on a communication level that needs to be worked out. It is very much like a group project, but all the technologies are there. It's just about now working out the pieces. And hopefully through this experience, both pull that all together, but then recognize that we need to sustain that going forward. I mean, it's one of these things that's very frustrating. I'm relatively even late to the game. I started working in infectious disease in 1997. There are people who've been working on it for decades more than that have, for many, many years been saying, this is coming, this is coming. And I always think to Laurie Garrett and The Coming Plague, one of my early introductions to this, and Richard Preston and other, writers who've been saying for years, we should be ready. And fundamentally, they all know that infectious diseases kill more people than any modern war, and most of our historic wars, even the Great War, infectious disease is always the major killer, and it can destroy economies. And if we invested just a fraction of that, we would be in a very different place.
Part of that investment is, as you said, we hear from public health leaders that we have to expect to live with the threat of new infectious disease outbreaks. You and your team are working on a way to potentially detect some of these new pathogens that come up, new viruses and bacteria that might cause disease among human populations. Can you explain how that works?
We just most recently published a paper in Nature that describes a technology called Carmen. And it's essentially pairing CRISPR technology with microfluidics or other forms of miniaturization, in this case microfluidics. But this is basically miniaturizing these tests so that you can run 64,000 assays in parallel. And we do everything in replicates, so we can run almost 5,000 tests at a time. And this allows us to scale up greatly. When we originally developed it, we were more focused on the ability to test for lots of different viruses, take one patient sample. For COVID, I think I've seen different numbers, but when people do testing, maybe 10% of them are COVID, and there's this other 90% that we don't know what it is. And it's frustrating. I mean, why is it that we always leave and we don't know what we're infected with? But we would be able to pick up these things and be able to start really understanding them and studying them if we were constantly looking for lots of different viruses. So this particular test we showed could detect 170 different viruses that are known to infect humans, but we anticipate using it and moving quickly to get it out into the world to be able to test for COVID in that, over 1,000 samples or more than that. and then also at a time, and then to also test for other viruses that could be co-circulating.
And how do you anticipate that being used? Like, would people be tested on a regular basis, or just blue sky, how you would like to see something like that being put to use in a public health setting?
For the last 6 years since Ebola hit, we have been fine-tuning and honing how we think things, we would like to put forward. And we created something with my collaborators in West Africa, and Christian Happy is my lead partner there, but a whole team of individuals working on this, created something called Sentinel. And we were just actually awarded, just as COVID is hit, secretly circulating, we were awarded the Audacious Prize through TED to create something called Sentinel. And the idea is that we have point of care tests, like paper-based tests that can happen in the field or could happen in your home, paired with the kind of test I was describing that could happen in hospitals, where if you don't, if you test at home and you don't have or test in your local clinic or with a healthcare worker and you don't have the obvious things, you can then go into the hospital and see if there are a wider range of viruses you might have. And if not, your sample could be sent to a sequencing center where we look to see if there's anything else we're not picking up. And so this sort of idea of Sentinel is really having different types of ways of detecting at every point in the healthcare system, and then connecting that information in real time across a grid. So we've been developing cloud-based systems that we can share genomic data and diagnostic data in real time, paired with mobile applications with great organizations like Demagi and Fathom, who've been working in this space, creating these kinds of technologies that allow all this data to be shared. And so that is actually, now when we say it, it's like, well, duh, you need this, but we've been working on it for a while. We're excited, though. There are a lot of other folks now, a lot of folks that were there and new people who are here. So it's a great ecosystem, and we're finding new partnerships, and we're excited to get this out into the world.
And key to that, as you said, seems like a way to really connect and network the entire global health community. So I'd like to get your perspective on the question of global collaboration. You referred to it, and infectious diseases and public health are really increasingly a global issue, where what happens here in the US can very quickly affect people in Asia, Europe, Africa, and vice versa.
Yeah.
How strong, in your opinion, is the global public health infrastructure?
Well, I mean, it's fundamentally a lot stronger, but it's also going through growing pains. I mean, one thing, could you imagine this situation back in 1918 or even 20 years ago, when you didn't have Zoom, when you didn't have, Slack and all of these different ways that people are engaging with each other and share, when bioRxiv didn't exist, where people were publishing papers in real time. So that is really exciting, is the way that there's this connectivity and this cooperation that can happen. The healthcare system still have a lot of things to work out. I mean, we know we've been talking about electronic medical records in this country for years, and the fact that every hospital has its own system, and I don't have my medical records from middle school or high school. I mean, it's nuts. There's a whole lot that we still have to fix here. And so sharing across different institutions is still a problem. There's a lot of things that need to be worked out. And infectious disease is a place to really think about it, because that network matters. The connectivity matters of a lot. So we need to be able to share data in real time in medicine in general, but nowhere more necessary than infectious disease, where it's a volatile situation that's moving in real time that people need to share. And we see this, and we're actually one of the slow. We've been publishing very slowly in this outbreak, even though we're the ones sort of saying publish early, and fundamentally because we're actually looking at all of our data and checking it every which way to Sunday. My partner Jeremy Lubin saw that mutation that everyone's talking about now, the D614G mutation that might or might not, affect the virus months ago, and he's been working on it diligently for months. We're not publishing that stuff right away because we are afraid of saying something too early. Like, it's a dance we're trying to figure out. I don't want to publish bad data. I don't want to, and that is something that we're seeing. We're seeing this publications are happening on Twitter, on bioRxiv, and some of that is great and some of that is dangerous, because we're getting a lot of misinformation. We're getting a lot of things going sideways. And so that is the interesting dance that we're learning about here of, how fast should you go in sharing information? How cautious should you be? How do we communicate with each other in this age of misinformation and alternative facts? How do we do that, I think, is one of the big challenges.
And in the midst of responding and trying to learn about a virus that is so new, in the midst of all that chaos, what gives you hope?
I think it's just every person I see that's just trying. You know, at the end of the day, there is chaos. There are also bad actors. I want to be honest about that. But you, Alice, I mean, frankly, you give me hope. Like, you're a terrific human who's trying to tell good stories, and having, you know, getting to engage with them is just phenomenal. And I always do shout outs because although I miss a lot of people, but when I was just talking about that, all the chaos and an outbreak, like my partner, Lara Salahi, who wrote a book with me called Outbreak Culture that talks about how it gets so toxic. I mean, we're working together on a project, and like, she gives me hope. So many of the people, the students I work with, they give me hope. To me, my motivation also comes every morning. I wake up, I swear I almost like, I have this feeling of tension, of thinking of every child trapped in a home that they can't get out of, you know, because of social distancing. And social distancing, you know, was needed in this case to stop something that was a hemorrhaging situation, but it's a blunt instrument that is dangerous, and that we're seeing a lot more. You know, we're seeing so much devastation and collateral damage from that. And we're seeing a rise of suicides and overdoses and all of these other things. And you all talked about this in time as well. But to be honest, it's like, it's this kind of the opposite of hope, right? But it's that's the motivation. So it's, the hope is in seeing all the people that are trying, the healthcare workers on the front lines, every person playing their part. And the other motivation comes from help get those people, you know, out, get children back in schools, get everyone out in safe environments. So after all of that talk of all the technology is great, we're not doing great. We got to do way, way better than this.
Thank you, Dr. Sabeti, for providing us with that sense of purpose and fire, and for sharing your expertise in making sure that the next time we face a new infectious disease, we'll have the right tools and experience to confront it and hopefully minimize its impact on the world's health.